Welcome back. In this lecture, we will discuss the study of learning and behavior. In other words, the science of behavior. We will look at explaining behavior and how to find circular explanations. We'll look at how learning is measured, and we'll go over research designs for the study of learning. I want to begin this lecture with this quote from Thomas Huxley, which I think captures the spirit with which we need to go into the study of any science, especially one so close to us as our own behavior. So excuse me as I read it to you. My business is to teach my aspirations to conform themselves to fact, not to try and make facts harmonize with my aspirations. Sit down before fact as a little child. Be prepared to give up every preconceived notion, follow humbly wherever and to whatever abysses nature leads, or you shall learn nothing. It's a strong statement that we have to be prepared to give up our beliefs, our preconceived notions, in order to learn from the facts. But I feel that in order for us to learn and to conduct science, that is exactly how we need to do. What is the goal of a natural science? Well, essentially the goal is to understand, explain events or phenomena. So the goal then is to identify the physical events, the causes of other physical events. If behavior is the event that we're interested in, then we need to identify the causes of that behavior. So how do we go about explaining it? Well, let's look at an example. Here we see a rat pressing a lever. It presses the lever, and then it's ducking its head into a little food delivery dish. And someone may ask, why does the rat press the lever? How would we answer? Well, one might be tempted to answer that the rat presses the lever because it knows it will get food. And the question is this a scientific explanation? We use explanations like this all the time for ourselves, for our friends, and for our pets. Why does the pet do something? Because it knows I'm about to feed it. Or because it knows such and such is going to happen. That's all well and good on a day-to-day basis, but when it comes to science, is that a good explanation? The answer is no. These are not scientific explanations. Instead, this is a circular explanation. It's circular because the cause, in the case of the rat, the cause is knowing that it will get food. That cause is inferred from the very fact that we're trying to explain. The fact that the rat is pressing the lever. What do I mean by this? Well, let's walk through it. What is it that we're trying to explain? We're trying to explain why the rat pressed the lever. And our answer is that the rat knows it will get food. Well, how do we know that the rat knows it will get food? Because it pressed the lever. That's a little bit of a problem, is it not? Essentially, what we're doing is we're saying the rat pressed the lever because it pressed the lever? I mean, the only way we know that it knows that it pressed the lever is that it actually pressed the lever. So this is an example of a circular explanation. It relies on the fact that we're trying to explain to come up with an explanation. And so we're essentially just going in circles. In order to conduct a science, we need to avoid circular explanations. So again, the primary goal of behavior science is to explain behavior. We want to know why we do the things we do. So the questions then are, can behavior be predicted? Can we, if we have enough information, predict what someone will do or what an animal will do? And even more so, can behavior be controlled? Meaning, can we set up a situation and by setting the situation up in a particular way, get people or animals to do something specifically? Can we change things in such a way that it gets people to not smoke? Can we change things in such a way that it gets people to reduce drinking or to select healthier food items? Things like that. Those become the central questions. Can behavior be predicted? Can it be controlled? There are two main approaches to this. Two main approaches that are taken in psychology to understanding behavior. The first I'll call the common sense or psychological approach. It's the most common one. It's the one that we see everywhere. And this basic approach is that we look at the stimulus or the environment, and we see that as inputs into the participant, the organism that we're studying. And then we look at the response that that organism made. And the next step there that's generally taken is, so in other words, we might see that the rat is in a operant chamber, in a chamber where there's a lever and it can get some food. The rat being the organism, the response is that the rat presses the lever, and then our next step is to kind of say basically what we said on the previous slide, that the rat presses the lever because it knows it will get food. We make this inference about something going on inside the organism. As we just mentioned, though, this is problematic. Why is it problematic? Well, it leads to circular explanations. Another example, we see a child in a classroom. So the classroom is the stimulus, it's the environment. The child is the organism. The response from this child is that he is very active, is wandering around, is doing things that are disruptive, and then we infer from that that the child has ADHD. And then we use the ADHD as an explanation for the very behavior that we're trying to explain. A circular explanation. Instead, we need to use the natural science approach. This is a different approach that helps us to avoid making these circular explanations, or in B.F. Skinner's words, explanatory fictions. The natural science approach basically has a few assumptions that we'll walk through here. The first assumption is that all behavior is caused. That is, behavior is the result of experience, our physiology, our genetic makeup, and our biology, and the current situation. The next is that all causes must precede their effects, meaning that an event that hasn't happened yet can't be understood as a cause. So, for example, if I were to ask you, why are you enrolled in college? You might answer, well, because I want to get a particular job, and for that job I need the degree. Well, the degree hasn't happened yet. The job hasn't happened yet. Therefore, those future events are not causes for you being in college right now. They don't count because they haven't happened. Only things that have happened can count as causes. So instead, you'd have to look into your background and see what is it about your background that led you to college. What is it that who were the influences? What were the events that led you not only to college, but to this particular college, and not only to Georgia Southern, but to this particular class, right? And you could look at the events that led up to that, and those we could look at as causes. But things that haven't happened yet, well, they haven't happened, so therefore they are not causes. Causes must be natural phenomena. So, in other words, they have to be things that we can objectively measure and detect. So the soul, the mind, the id, the will, these things are all concepts. They're not actual things. We can't measure these things. We can't detect these things. These are things that are conceptual in nature, philosophical in nature, if you will. And lastly, we strive toward parsimony. The law of parsimony, it's also known as Occam's razor. Sometimes it's just abbreviated as keep it simple. Basically what we're going for here is the explanation that is the simplest that fits the data. So it's not just simple for simple's sake. It has to be the simplest one, the one that makes the fewest assumptions, while still explaining the things that we've observed, while still accounting for the facts. So we want it to be a simple explanation, meaning that it makes the fewest assumptions necessary. But if it's oversimple, it's simplistic, then that means that it is really going to fail to account for things and also not what we're going for. It's the simplest explanation that accounts for the data. Now, to objectively study behavior, we need to define it. We need to be able to categorize it. We need to be able to state what counts as a behavior and what does not then count as a behavior. And the way we do that is through operational definitions. We define a phenomena, whether it's the stimulus or the behavior, in terms of how it's measured. So this is kind of like writing a recipe for the stimulus or for the response. So let's give an example. Here we have a picture of a rat pressing the lever. We want to define a lever press. So we may define that lever press as the depression of the lever with the force needed to complete a circuit. And if we know exactly what that force is, in this case 21 grams, then we could put that in there, and that would tell us exactly how hard the rat has to press that lever down in order for it to count as a lever press. Another example, a fear response. We could define a fear response as a rise in heart rate, respiration, or as a change in the electrical conductivity of the skin, or GSR, galvanic skin response. And the figure, the graph, shows how these changes might happen. On the left side, we see essentially steady state or baseline for respiration, galvanic skin response, blood pressure, and heart rate. And we see that everything is essentially level. Where the blue line drops down is where the fear stimulus occurs. A loud noise, something like that. After that, we see, starting at the bottom with heart rate, we see heart rate increases. Right? The peaks are closer together. We see blood pressure rise, and we see in the galvanic skin response, we see an increase there as well. So all of those would be measures of the fear response. Why is this important? Well, this is important because the behavior can vary a lot in how it looks. So, for example, when the rat's pressing the lever, it could use one paw, two paws, it could use its snout, it could bite the lever, pull it down with its teeth, it could stand up and jump on top of the lever. As long as the lever is pressed down far enough to complete a circuit, it counts as a lever press. It doesn't matter, according to this definition, it doesn't matter how the rat did it. All that matters is that the lever got pushed down far enough to click. That's the power of having operational definitions, is it allows us to categorize what behavior counts and separate it from the behavior that doesn't count. So if learning is a change in behavior due to experience, then that means we must be able to measure changes in behavior. So how do we do that? What are the measures that we can use? I'm going to go through a whole bunch of different measures fairly rapidly, so please bear with me. First, what we can look at is a reduction in the number of errors made. The graph on the right is showing the number of errors made across days that rats run through a maze. So we see 17 days, and on day 1, the rat is making over 250 errors when running through the maze. By day 17, it's down to making around 50 errors, or 60 errors. So that reduction in errors is a change in behavior, and we call that change learning. Another example is topography, or the form that behavior takes. Here what we're looking at is tracing inside the lines of a star, and on the first trial we see a lot of shakiness in the line. By trial 15, very little shakiness, and the line stays inside, showing a change in behavior. Intensity. Here we're just talking about how intense the response is. What's being measured on the graph is the intensity with which a rat presses on a lever, and we can see that on the graph on the left that the rat is pressing at most of the time at 29 grams, but when required to press harder, in other words, the tension on the lever is increased, so the rat must press harder for it to click, then we see that the rat is now pressing at between 41 and 45 grams most of the time. Speed. So how quickly is a task completed? In this case, the graph is looking at the average time to complete a maze. And we see that on trial 1, it takes about 23 seconds to complete. And by trial 17, it takes less time. They're able to complete the maze in around 7 seconds. Now, seeing that, we might, by looking at how fast they're solving a task, we might assume that they're making fewer errors. But we don't know that unless we record both things at the same time. So here, looking at speed, we see, yeah, they're getting faster at the maze. That doesn't necessarily mean that they're making fewer errors, but we know from our previous graph that looked at number of errors made that they are also making fewer errors. So by that would tell us by collecting both sets of data at the same time that they're becoming more accurate and faster at the same time. Latency is easy to confuse with speed. Speed is how long does it take to complete something once you've started. Latency is also known as response time or reaction time. So it's how long to start doing something once given the go signal. So once the stimulus is presented, how much time passes until the response happens. It's kind of like the time between when a stoplight turns green and you press on the accelerator. That is reaction time or latency. Lastly, rate or frequency. How often does one engage in an activity? And here we measure rate the same way we measure the rate of travel, for example. And like if we want to know how fast we're going, how fast we're driving, we look at miles per hour. In the case of behavior, if we're interested in rate, then we're looking at how many responses per some unit of time. Oftentimes what we look at is responses per minute. So, or in the case of the graph, letters per minute. So looking at typing, Morse code in this case. But this is a measure then of how rapidly a behavior can be completed over and over again. And if we combine rate with the measure of accuracy, then we get something called fluency. That is, we have both speed and accuracy combined. So with these basic measures, it allows us to look at or detect changes in behavior. In other words, changes in these measures are learning. Let's now look at some research designs. For those of you who are taking or have taken research methods, this may be a bit of a review for you. First, let's look at descriptive studies. What are they? Well, descriptive studies are essentially studies that describe characteristics of a population. These are studies that use surveys, questionnaires, natural observation, things of that nature. So let's look at an example. On the red axis, we see the percent of each of the countries who are obese. So all of the countries are along the bottom axis. On the green axis, we see percent of the population who walk, cycle, and or use public transportation. So we'll now put the data up for each one of these. So percent of obesity of the population of each of these countries, and the percent of the population who walk, cycle, and use public transportation. Seeing these two sets of data on top of each other, does anything jump out at you? Perhaps it might look like there's a relationship between these two variables. That as obesity is higher, or that in countries in which obesity is higher, there's also a lower proportion of people who walk, cycle, and use public transportation. What we can do, though, is we can put these two variables together on one graph and do what's called a correlation and see whether these two variables correlate. And if we do that, which I have done here, so now on the y-axis we have percent of those who walk, cycle, and use public transportation. On the x-axis, the red axis, we have percent of obesity. And now each one of the dots represents an individual country. Which one is the United States? We can run a correlation on these data now. And if we do that, we get a Pearson's R value, for those of you who have completed statistics. The Pearson's R value comes out at negative 0.88. And you may recall that that is a very strong correlation. What does that mean? Can we, from this, then state what the relationship is between these variables? So, for example, is it meaningful from this to ask, does the lack of walking, cycling, and public transportation in the United States cause obesity? Actually, no. That's not a meaningful question. Because when we use descriptive studies, we have not manipulated any variables. All we've done is collected descriptions and then put these descriptions together. But we don't know if there is any causality between these two things at all. In fact, they may not be related at all. And it would make just as much sense to ask the question this way. Does the level of obesity in the United States cause the lack of walking, cycling, and public transportation? Either way of asking the question is just as nonsensical, actually. It could be that the percentage of obesity in the United States has to do with the availability of good food, the price of food, things of that nature, and the low percentage of those who walk, cycle, and use public transportation is because there's a lot of the United States that's rural and we don't have a lot of good public transit. So in other words, the data here may not be related to each other at all. But we don't know that because all we've done is run a descriptive study. Experiments take us to the next level and allow us to determine causality. So what is an experiment? What makes it special? Experiments are studies in which variables are manipulated. The variable that's manipulated by the experimenter is called the independent variable, or IV. And the variable that is measured by the experimenter is called the dependent variable. With experiments, we can determine cause and effect. Why is that? Well, because we are actually manipulating a variable. We're turning something on and off. By turning the independent variable on and off, we can see whether it has any effect on the thing that we're measuring. So, let's look at an example. Will a rat respond more quickly, so press the lever more frequently, if one response produces a pellet of food? So the rat presses the lever once, gets food, presses the lever again, gets food, etc. Or if it has to press the lever 10 times to get one pellet of food. So it has to press 10 times, can get food, and then has to press 10 more times to get food. Under which condition will the rat press the lever more quickly? Or, in other words, have a higher rate of responding. So, here, what is the independent variable? What is the dependent variable? The thing manipulated by the experimenter, or the independent variable, is the number of responses required for food. What is the dependent variable? What is measured by the experimenter? Well, the rate of responding. How many responses per minute? So there are two ways that we could go about addressing that experimental question. Two different research designs. The first is called a between subjects or a between groups design. In this design, each group would get a different level of the independent variable. So let's look at our example. Also, groups must be equivalent for valid conclusions, and generally what we do to make sure the groups are equivalent is we use random assignment. So say we have a total of 10 rats, and we want to know whether pressing the lever 1 time for food versus 10 times for food produces faster responding. Then we would, of our 10 rats, we would flip coins and randomly assign 5 of the rats to the 1 response group and the other 5 rats to the 10 response group. We would run the experiment. Maybe we'd let the rats work under these conditions for a number of days. And then we'd look at the results. We'd see how many responses per minute the 5 rats in the 1 response group did versus the number of responses per minute the 5 rats in the 10 response group did. And we would test those rates for sameness. In other words, we're looking to see our statistical tests assess whether the two groups are the same. Which type of statistical test would you use on this? For those who have completed stats. The other approach is a within subjects design. In this case, each participant or each subject gets all levels of the independent variable. We don't have to worry about groups being equivalent because, well, they're all the same rats in this case, right? Everybody is in all groups. So we're just comparing each individual to itself, essentially. So we're going to look for changes in each participant as they experience the different levels of the independent variable. So the example here then is we would have 10 rats. All of them would work under the condition of 1 response gives them food, and all of them would also work under the condition of 10 responses gives them food. And we would then compare the rate of responding in the one condition with the rate of responding in the other condition. Which of these two approaches is correct? Which is the best approach to take? Well, both of them are actually equally valid approaches. They're both really good. There's not a wrong answer. What is important is that one has proper controls and things like that that get into details that are beyond this particular presentation. But actually, both approaches, both of these designs, are excellent to address this question. Another type of experimental design, potentially one that you've not heard about before, is called an ABA design. The ABA design is a single subject design. In other words, we're looking at just one individual and we're conducting research on this one individual. It might be a pet, or it might be a client, it might be a child with behavioral problems. In other words, this is something that is used a lot in clinical practice. The goal here is to record behavior before treatment, during treatment, and after treatment. So the goal is to see whether the treatment had any effect. So what does the A stand for? The A stands for baseline. In other words, this is the level of the behavior prior to any intervention, prior to any treatment. So on the chart, on the graph, on the y-axis it reads appropriate play acts per minute. So what we're looking at here then is a child when interacting with peers. We want to see how many times does the child interact with peers in an appropriate way, like share toys, etc. This would have to be something that is operationally defined. And then we would look for examples of appropriate play acts over the period of recess, the period of observation. In this case, we see that the number of appropriate play acts for this child are very low. So over these minutes of observation, we see that there are only a few appropriate play acts. Once baseline has been completed, so we see this is what the child is doing in the absence of treatment, then we introduce treatment, or the B condition. And the treatment might be that someone, it could be anything from giving stickers for playing appropriately, for giving praise. It could be any number of things that the treatment might be. In this case, though, what we see is that when the treatment then is in place, so under condition B, we see that the number of appropriate play acts, like sharing, increase. Now, we want to make sure that that change in behavior isn't due to something other than treatment. Right now, what we've seen is that the number of play acts, appropriate play acts, was very low, and then it changed and went up. But that could be for any number of reasons. Even though we started treatment and it looks like the treatment may have caused that change in behavior, we don't know that for sure. The way we can be more sure about that, though, is we go back to baseline. We stop treatment and we go back to the way things were before. And in this case, we see that their number of appropriate play acts decreases back down to baseline levels. That tells us, that suggests to us, that the treatment was the thing responsible for the change in behavior. But again, we still don't know that for sure. The way we can become more sure is we reintroduce the treatment. And if we reintroduce the treatment and we see behavior go back, so now they're engaging in more of these appropriate play acts, that makes us even more confident that the treatment is the cause for the change in behavior. This is a very powerful way of assessing treatments. It's also a very, very powerful way of determining whether a treatment is causing the change. In other words, being able to explain why behavior happens. So this is a very strong design. It's very useful in the clinical field, particularly when working with children or pets, but in general in the clinical field. And it's powerful because each participant is really just being compared to themselves. We're not comparing them against other people. We don't have to worry about any group differences. We're just looking at the behavior of this one individual in comparison to themselves. So we can have very high internal validity with this approach, meaning that we can have a very strong sense that the thing causing the change is the treatment. Why are experiments so powerful? What is it that makes them powerful? Well, they're powerful to the extent that we have control over variables, that we can rule out alternative explanations, and we can determine that the independent variable, the thing that's manipulated by the experimenter, is the thing that's controlling behavior. There are, of course, criticisms. One is that these experiments are generally conducted in very artificial situations, and that is true. Oftentimes they are. But experiments have to be done that way. That's part of controlling variables. We need to rule out alternative things, and the easiest way to do that is to create a somewhat artificial situation in which those other things aren't present. The way we then address that artificial nature of experiments is we have to conduct both laboratory research and field research. So once we've done some work in an artificial setting, in a very arguably sterile setting, then we try to take that into the real world, develop technologies from that science, and see how well they apply in the field, in the real world. What this allows us to do is test whether the results from the lab generalize to the real world and generalize into these other settings. To summarize, the goal of science then is to predict and control events. We want to determine through this prediction and control the causes of these events, and in particular in this case, the causes of behavior, why we do the things that we do. While doing this, we have to watch out and not fall prey to circular explanations. These explanations are so prevalent in our daily lives, it's easy to miss them, but we need to make sure that we don't fall prey to them. We need to define our measurements and our behavior in a way that allows us to detect learning, that is, detect changes in behavior. By using descriptive studies, that may allow us to make predictions about behavior, make predictions about what someone will do, but not necessarily explain what they will do. Experimental studies allow us to make both predictions and potentially have control, essentially establish causality. And as we can establish causality, control, that moves us forward in explaining why we do the things that we do.